Hello and welcome to today's webinar on eDNA. It is certainly an exciting emerging field, and we have done our best to get you a panel of expert presenters together who are on the cutting edge of this science. I'm your host, Cray Price with the International Water Training Institute, and I'm pleased to welcome not just our presenters today, but each of you, the attendees from all around the world. This will be an interactive discussion today, and given the diversity and where we're all coming from around the planet, I'm sure we'll all learn from each other and maybe even find some new applications that we might not have thought of. Now, if you could all, the presenters, if you could turn your cameras on and let us know who you are, where you're coming to us from today, what you do, and how you got into this business of eDNA, and also how long you've been doing it. I guess that's a lot of questions, but we're just thrilled to be able to welcome each of you on your inaugural first Australian Water School webinar, which certainly won't be the last because we've already got some upcoming training scheduled, and so we'll be announcing that throughout the webinar today. So let's go in the order we see it in here. Josh, if you could give us a quick introduction.
Yeah, g'day, Cray. My name's Josh Griffiths. I'm a wildlife ecologist. I actually specialise in platypus ecology, and so that was my avenue into eDNA. I've spent a lot of cold, wet, and lonely nights trying to catch platypuses. And eDNA has kind of revolutionised how we can monitor those populations. So that was in the very early days of eDNA in Australia. So I've been working in this field now for about 10 years now, I think.
All right. Well, thanks for that. And now I know, I always wondered what was the plural of platypus, and was it platypi? Maybe not. Emma, over to you.
No, thank you. Hi all. So I'm Emma Walker. I'm at eDNA as a project manager. I've been doing that first. How I got into eDNA was I did my honours in eDNA. Was very fortunate to work on looking at the impacts of the Black Summer bushfires through the east coast of Australia, and we were really lucky to have pre-fire data in those areas, so we could do a really nice comparison across the bushfire affected areas. So I've been working in eDNA now for around just over 3 years. And seen a wide range of applications across our different projects, which is great.
Excellent. Thanks for that introduction. Halo, over to you.
G'day, my name is Halo. I'm a molecular scientist at EnviroDNA. I first got into eDNA while I was doing my PhD in actually a completely different field of research. I was doing it in human health pathogens. But I was doing genetics work there, and I was looking for work while I was doing my PhD, and the folks at EnviroDNA took me on as a casual at first, and I just really fell in love with the technology and all the things we could do with it, and how much we can surveil using eDNA technology. And so once I finished up my PhD, I just continued on this path.
Excellent. Well, we're looking forward to some innovative science here that we'll hear about. Let's have a look at the poll results. Thanks for filling those out. It looks like we're pretty much split. One person says they know more than the presenter. That's great. But we're pretty split between complete newbies, those who have heard of it, and some who are keen to learn more, but already have some basic knowledge. So that's good to see. And I think if we had a poll like this next year, maybe we would see a little more experience. And so that's what we're aiming to bring you. It looks like we're pretty split across the board in terms of what people want to use it for. Emma, just any comments that you might have on that? Any surprises there? Is this what you would have expected to see out of the poll results?
No, I feel like that's, fairly the kind of spread I would expect. Just based on as well the different people that we've worked with and their knowledge.
And have you seen this increasing? Like if we'd had this poll a year ago, say, has this been accelerating the interest and the exposure that the topic is getting?
Yeah, definitely. It's so great to know, see people being more interested in it and, develop that knowledge in the different uses and how they can apply it to their projects or, their areas of interest. So, it's really nice to see.
Perfect. Let's learn a little more about it. I'm a learner as well. This is a brand new field for me, and I'm very excited to hear what Josh has to say about it. So we'll see you shortly after the presentation.
All right. Thanks for that intro, Cray. And good afternoon, everyone, or good morning, good evening, depending on where you're from. I'm coming to you today from Gunai-Kurnai lands in eastern Victoria or southeastern Australia for people that are overseas. Really excited to present this today, and I think the poll results probably highlight the diversity of how eDNA can be applied and across different organisations and different areas and industries, which makes a thing like today quite challenging. We can't cover everything, but hopefully I'll be able to give you a good background knowledge. So today is actually the first part of a series of workshops that we're doing with AWS. So today's very much an introduction to eDNA. It's very much targeting, beginners or people that are just starting out on understanding about eDNA, want to learn how they might be able to apply it in their field, but probably not using it at this stage. Our upcoming workshops that are happening over the next few months, we'll delve a little bit more deeper into some of the details around eDNA, some of the technical aspects, and I guess how you can apply it more effectively, and I guess understand the outputs that come from it and how that data might be analysed. So certainly have a look at that course if you're interested in learning a bit further. As part of that, we're also going to be sending people out some water sampling kits. So you'll be able to go and take some water samples from your local waterway, and we'll do a vertebrate analysis, which you will learn what that involves in a little bit. So as we saw from the polls, eDNA is an incredibly widely applicable and broad topic. So it's difficult to cover everything in a one 45-minute webinar. I guess what I'm trying to do today for those beginners is really give you an understanding around what eDNA is, the different types of methods that are used, and to get you to start thinking about how it might be applied for your area of interest or your work. So let's get things underway. So just a little bit about EnviroDNA. So we're a small private organisation. We're based in Melbourne. We have dedicated eDNA labs specifically designed to process eDNA samples because they're quite unique and require some careful considerations. It says here we've got 25 employees. If we do, I'm not sure I've met half of them. I would say we've got about 15 or 20 employees, but we work with a lot of different clients across a range of industries, mostly in Australia, but also abroad as well. So I guess my role within EnviroDNA, so as I mentioned before, I'm a wildlife ecologist. So I've spent almost a couple of decades using traditional techniques, whether they're observation, trapping, counting. And so it kind of puts me in a bit of a unique position, having seen both sides of the coin. So my role these days is really that link between all the technical stuff that happens in our lab, and people like Halo do an amazing job in that space. But geneticists also tend to talk in their own language. So it's up to me to sort of translate that to the end user who often don't have that technical knowledge. So hopefully I'll be able to do some of that for you today. So first of all, so what actually is eDNA? Essentially, it is every organism is shedding genetic material into its environment, and as soon as it gets shed, it becomes environmental DNA or eDNA. That comes from a range of different sources. So it might be epidermal cells like skin or scales or hair. In water, aquatic systems, which we're going to focus on today, it's largely from urine and feces, from waste matter. So probably not a pleasant thought if anyone's planning on swimming in their local river this weekend. But all of that contains DNA, and we can now take environmental samples, whether that's a little bit of water or some soil or even air, and we can now extract and identify these tiny traces of DNA. So this is something that wasn't really possible, 10 years ago, but now our genetic techniques are becoming a lot more sensitive and a lot more powerful, and also a lot more economical to apply. So we can detect these tiny traces with a lot more precision than we could previously. Now, I'm sure everyone that's online today has seen a CSI-type TV program where the detective goes in and swabs the crime scene and uses that to then identify the criminal. Essentially, we are using very similar techniques in the wildlife space, but instead of identifying individuals, and I know this is one of the questions that will come up today, we're not identifying individuals, we are identifying species. And the reason is that once DNA is in the environment, it degrades and fragments very quickly. So we're looking at tiny traces of DNA, fragments of DNA, and being able to identify species from that, but we don't have enough data to be able to identify individuals. Now, we're really going to focus on water today, and it's probably the most common use of eDNA, whether that's freshwater or marine. I would say today I'm going to focus on freshwater ecosystems, but the same, everything that I talk about applies in marine systems. And also the same techniques can be applied across terrestrial systems. So there's a range of different samples we can look at from water, as I said. Scats are actually really good. So we've got some amazing and detrimental predators in Australia that are very good at going out and collecting a lot of our native species and then leaving us a little sample that we can analyse. So that's actually really good in terrestrial systems. We can look at soil, and soil is probably more used for something like ecosystem health measures. So looking at bacteria and fungi as a diversity measure for ecosystem health. We work with people that might swab surfaces, whether that's leaves or tree trunks or hollows, to see what's using those things. There's a whole field called eDNA, which is DNA that's found internally in things like mosquitoes and leeches and some predatory flies. So we can actually analyze that to see what they're preying on, which is a project that I really want to do at some stage. And even looking at, petals of flowers to see what pollinators are visiting those flowers. So there's a real range of techniques that can be applied. As I said, very much going to focus on water today, but just want to highlight that it can be applied across a range of different systems. One of the challenges when we get away from aquatic systems is really being able to detect or find the DNA in the environment, because it doesn't move, it doesn't flow like it does in aquatic systems. An emerging field that is coming out is something called air DNA, where we're even able to extract fragments of DNA from the air, which is pretty exciting. So I think the one thing to recognize here is that eDNA is simply another tool that we can use. It's another monitoring tool that we can use. It has some really good advantages, but also some limitations. So it's about understanding what's most effective for your ecosystem, but also your target species, and what are the questions you're going to answer. So it sits in this, call it our wildlife survey toolkit. And so today is all about understanding what eDNA can and can't do so that you can make those decisions to apply it or not, or use it in combination with other techniques, which we find is really powerful. Now, you might have noticed from the title of the talk today, Beyond Tracks and Scats, the reason being is that eDNA is simply another trace that's left behind by organisms. So for decades and for probably hundreds of years, we've looked at animal tracks and animal scats to infer presence. Essentially, we're just doing that, but at a microscopic level. So it's really a more, technological advanced version of what we've been doing for many years. Some of the advantages around eDNA is, I think first and foremost, and for those that sign up and will get sent some water kits, is that the sampling methods can be incredibly easy. There's different ways that you can sample waterways and collect and filter water, but at its heart for most of our projects, it's a very simple process, which means that you don't need specialized equipment, you don't need trained professionals to go do it. Most of our projects, we end up sending sampling kits out to people. They do the sampling themselves and then send them back to us for analysis. It's also been really powerful for us to run citizen science projects, and I'll highlight one of those in a minute as well. So the actual sampling methods, provided a little bit of instruction and you follow some protocols, they can be really simple to do, which means basically everyone can get involved and really take ownership of their projects. Now, for anyone that works in the wildlife space, having a technique that is non-invasive means that you don't have to write animal ethics permits, which I'm sure everyone will be happy about. Of course, it also means we are not disturbing the animals that we're targeting. We don't need to trap them. We don't need to observe them or disturb them at all. And not just our target species, but also any bycatch that we might get from traditional methods. So it's not invasive. Probably the key benefit of eDNA is that it can be incredibly sensitive. So, for example, for our, I work with Platypus a lot. We can go, for most waterways, we can go and take a couple of water samples, run a couple of tests on that in the lab, and we get over 95% detection rate. So very high confidence of detecting species if they're present, and conversely, if we don't detect them, we have high confidence that they're absent. So this is really important for any sort of monitoring technique, and usually it's kind of an order of magnitude more sensitive than a lot of our traditional techniques. Part of that sensitivity and advantage is that it doesn't just detect adults and grown individuals. We can detect things like fish eggs, we can detect gametes, we can detect juveniles that are really hard to physically identify. So we can pick up species across their entire life stage, which is incredibly beneficial. That high sensitivity means that we can detect species at very low abundance. So whether they're incredibly rare, which is a lot of our threatened species that we might be targeting, or it might be early incursions of invasive species, or simply species that are maybe cryptic or elusive that, avoid our traps. And I would say platypus is one of those. So we can use this to detect those problematic species that we haven't been able to get good data on previously. And it actually, where eDNA evolved from was actually around that detection of invasive species, because of course, if we can detect them earlier, we can employ methods to control and eradicate them a lot more effectively than when they're already established. Also, with a different technique that we'll talk about in a minute is that we can not just target single species, but we can get biodiversity estimates of entire taxonomic groups, or ideally from total biodiversity from an area. And we can do that from the same samples instead of having different methods to target different species. And importantly, we can now do things at massive scales over long terms because of these advantages, it being cost effective, it being easy to sample, and relatively cheap to generate this data. So just mentioned before, so these are the sort of kits that we'd be sending out. Essentially, it's a large syringe that's your water sampling device. That water then gets pushed through a very fine filter, which is this here, which captures all the genetic material that we want. And then this is what's getting sent back to the lab. So we're not transporting litres of water across the country or internationally. We're sending these simple little filters back to the lab for processing. So there's huge advantages there. Of course, for people that are doing lots of eDNA, you can use these sort of more, professional level pump and filter systems. There's some great advantages to that, but of course, there's also an investment to it. But yeah, if you are doing lots of sampling, they are incredibly effective and enables you to sample large volumes of water, which is a bit more difficult with your syringe. One of the things I just want to mention on, and we're not going to touch too much on survey design today, but I often get people coming to me that completely, forget their scientific training and anything about experimental design. As with any method, your survey design is critical to answer your question. Now, there's a few things that are, unique to eDNA that need to be considered, but generally that idea of replication and repetition is really important. So understanding what your question is and then designing your survey effectively is incredibly important. Okay, probably the one thing that I do want to take everyone to take home today is really the difference in these two different methods. So essentially, when we're talking about eDNA methods or eDNA detection, there are two main methods that are used. And so one is this target species detection, where we are looking for a single species only. And then we can also do these biodiversity assessments or multi-species detection, where we can try to detect multiple species from within taxonomic groups. Now, I try to sort of bring this back to methods that people might be used to. Now, I'm a birder, and so I spend a lot of time out in forests looking for birds. I think of the target species detection as maybe a cool playback survey. So you're only detecting a single species, but you can do it really effectively. Whereas the biodiversity assessment would be me sort of wandering through a forest and trying to identify every bird that I come across. That's a poor metaphor, but that's sort of how I think of it, and hopefully it resonates with a few of you. So I'll start by talking about the target species detection. This might be the thing that people are most familiar with. So essentially what we're doing is that we have a genetic probe and techniques that are designed to detect a single species. So that can be a range of different species, microscopic to macroscopic. It can be viruses, diseases, all kinds of different things. But we are looking for a single organism. The process is we go out and we collect our environmental sample, whether that's water or soil or whatever it might be. It comes into the lab and it goes through a DNA extraction process where all of the DNA in that sample gets extracted. So it's these colours represent a range of different DNA from different organisms. It then gets a species-specific probe added to it, and it goes through a process called qPCR. So that probe will only bind to its matching DNA, and then essentially that is what we're able to detect in our process, and then we can map the results, whether they're negative or positive, throughout the landscape. And so this process is relatively... Halo, I'm sorry, but I'm going to say that it's relatively straightforward and simple. I know it's not, but I guess it's an easy way for people to understand it. It can be done quite quickly. It's pretty cost effective and, importantly, very highly sensitive. There's also a quantitative capacity here as well, because we're able to quantify the amount of DNA in our sample, and that can be correlated with species abundance, although there are a lot of things that influence that. So just to illustrate that a little bit further, this is a range of DNA that we've extracted from our environmental sample. Here we've got our target species DNA. So this is the common galaxid, Galaxus maculatus, which is a fairly common little fish species in Australia, particularly southeastern Australia. So we have a species-specific probe that is designed to detect a unique region of the genome of that species. It also has a little fluorophore attached to it. When it goes through the qPCR process, it finds its matching DNA in amongst that sort of soup of DNA. And when it does, that little fluorescent signal goes off. And that's what we're actually able to measure in our process. So the intensity of that fluorescence allows us to understand how much DNA is present in those samples. But importantly, we're not getting any information about the DNA itself. So we are really reliant on that genetic probe binding to its matching DNA and nothing else. So it's really critical that that probe is designed and validated effectively. And so we want it to target a unique part of the genome for our target species. We want it to not bind to any other species that are known from the area. So it needs to be checked across all of those species as well. And we want to understand that it's sensitive. So we can design things in silico that might look good, but then when they're applied in reality, they don't work as well as they should for a variety of reasons. So understanding the sensitivity of this test is really important, and there's limits of detections and limits of quantification that should be calculated during good probe design. So at EnviroDNA, we've been doing this for a while now. I think this slide's actually a little bit out of date. I've got the feeling we're up to about 90 plus different assays for species across a range of different. There's my little crit up in the corner there. There's a range of frogs, great white sharks, a heap of different fish species. We've got a couple of reptiles there as well. We've certainly used this for a few viruses and fungus, so things like chytrid, which is important for our frog species. So we have a range of different assays that we've used and implemented. The other thing to consider when you're looking into using these assays is that we need to make sure that assays are specific for your particular region as well. So because there's some genetic variation within species across their range, an assay that might have been developed in the southern part of their range might be slightly different to one that's required in the northern part of their range. So understanding the spatial applicability of assays is really important. And I guess that's what you sort of chat to us about. So to give you an example of what we're able to do with this, so I mentioned before that we can do things at scales that we've never been able to do before. So I've been chasing platypus now for almost 2 decades through trapping and catching them and even observations. It's a very time and labour intensive process. A couple of years ago, we had a program called the Great Australian Platypus Search, where we were able to map platypus distribution across the entire state of Victoria. So These markers represent all of our eDNA survey locations. There was about 2,000 or just under 2,000 across the state. Green is where we detected platypuses, grey is where we didn't, and yellow are possible detections, really trace amounts of DNA that might indicate presence or not. But essentially, we've mapped an incredibly difficult species to monitor in the wild across an entire state. And this was really a 90% driven citizen science project. So we had volunteers going out and taking these samples for us. We had the time, we actually had lockdowns during COVID as well, which was an additional little challenge. But these are the kinds of things that we're able to do. And with that kind of sample size, you can overlay all kinds of different environmental variables to try to understand what's driving those distributions. And you can probably see just from this screen, there's some very clear patterns of detections, with high detections in some areas and really low detections in others. So, ultimately what we're trying to understand a lot of the time is what's driving those spatial patterns. And so we now have sample sizes to be able to do rigorous analysis to understand some of those things. Another species here at a much more local scale. So this is an endangered fish species, the Australian grayling. It's a diadromous species, so it lives in freshwater systems for most of the year in the sort of middle and upper reaches of waterways, but then it has to migrate down to estuaries to breed before then moving back up to its freshwater systems. And so this was a monitoring program we ran over a couple of years where we can see this is a sort of nice, relatively natural system where we can see the grayling are detected right along the system at different times of the year, as opposed to some of these other systems where they're only reaching the lower reaches and no further. Now, the reason is that there's some fairly significant barriers in those areas. So for a waterway manager, you can then go, one, you've got very clear evidence to know what the problem is. You can look at these barriers and work out, well, can they be removed? Can they be modified? Can we install something like fish ladders? And then importantly, once that management action is undertaken, we'll have the capacity to see whether grayling are then moving past and whether those management actions are effective. So, variety of different ways it can be applied. Typically, we're generally trying to understand species distribution at scale. And so there's a couple of ones at very different scales. I mentioned before eDNA being very good at detecting incursions of invasive species. It was what it was originally designed for. And so many of you will be familiar with this little critter here and the problems that it causes in Australia. And we've been running programs with a number of different organisations to monitor their invasion fronts, particularly targeting, high-risk areas. So whether they're a port system or a transport hub, where there's potential for incursions, it might be tourist hotspots in particular times of the year. So we monitor particularly around campgrounds and high visitation areas around Christmas and Easter when there's a lot of movement going around. And so the chance of introducing these species is pretty critical. And as I mentioned before, if you can detect them early, you've still got capacity to control and eradicate them. Once species like this get established, it is almost impossible to fully eradicate them. However, we can also use eDNA to track how effective those eradication efforts are. And I guess one of the advantages around using it for amphibians like this guy is that we're not just detecting the adults, which are often pretty visible and easily seen, but we can detect frog spawn and their tadpoles as well that are a lot more cryptic and not as easily identified. On a similar vein, we did a project a few years ago looking at incursions of particular species of mosquitoes from around the world. Again, looking at those high-risk areas, so airports around Australia where there's actually evidence. We know that these species are being transported on these planes. What we were trying to understand was, were they actually establishing? Could we find breeding populations around airports where there's lots of pools of water, stagnant water, whether it's a little drain, a wetland, a puddle on the side of the road, lots of areas where these things can be breeding and establishing and then spreading into Australia. The importance of these species was that not only are they an invasive species, but they are one of the key vectors for several diseases. So things like dengue and malaria, and at that time, particularly Zika, was of concern. Thankfully, we didn't detect these species at any of the airports, but we certainly found breeding areas of native mosquitoes. And so we were then able to advise the airports around management actions they could take to reduce the chance of these guys establishing, because as I said, we know they're coming in on planes, but they don't seem to be breeding at this stage. A couple of other things. This is a little bit different, and I guess a very sort of specific use, but I just wanted to use it to highlight the different uses. This guy washed up in near Portland, in Victoria, I think last year. We got swab scent of its bite marks. And similarly, this is an endangered plains wanderer that we're able to swab the carcass and the feathers that were found. And the goal there was to understand what was predating on these species. And to my knowledge, I think this was the first documented case of a orca predating on a great white in Victorian waters. And this was a fox or a couple of foxes demolishing a reintroduced population of one of our critically endangered birds. So again, using that really high sensitivity to be able to track the, I guess we're looking at the criminal, we're back to the crime scene again. On a similar vein, we often get scat sent to us, trying to identify whether it's is it a fox scat or is it potentially a endangered tiger quoll. So we're able to swab the outside of those scats and identify who it belongs to. That's very effective as well. We can also look at the interior of the scats and see what things are eating as a slightly different technique. So that's a few different examples of how we can apply that target species detection or our qPCR detection to look for single species. Now I'm going to move on to our biodiversity assessments. It's a very different technique. I guess at its heart, we're still looking at species detection, but it's important to understand, I guess, the differences between them when it comes down to interpreting the data and making that decision about which technique you're going to go for. So as I mentioned, when we're looking at our biodiversity assessments, it's using a process called metabarcoding. And what it does, it's able to detect multiple species from within a taxonomic group. And that can be quite targeted, or it can be very broad. So we can look for fish or decapods, or we can look broadly for invertebrates or vertebrates, even plants. There's a range of different assays for different groups that we can look at. The process at the field end is much the same. So the water sample collection still remains the same. How you design your survey might change because you're targeting multiple species that are going to have different habitats and different behaviours, but I guess the water collection process is the same. The DNA extraction step remains the same, and then things diverge quite widely. So we use the assays for a particular taxonomic group to build what's called a library. So what it does, those assays will be able to target, say, all of the fish DNA within our sample or all of the invertebrate DNA, and then it goes through a sequencing process. So instead of just a probe binding and saying, hey, I'm here or I'm not here, what we get back is lines and lines of genetic code. And we're talking about millions of lines of code here. Each of those lines represents a genetic sequence. And it goes through a bioinformatic process that, there's a QC and a cleaning process, so it gets rid of any junk or anything that doesn't meet our strict QC criteria. But then what it does, it clusters those millions and millions of lines of code into similar sequences. So this is a very, very simplified version, but we can see that these blue diamonds up the top, they're clustered because they're all the identical sequence. And same with the blue, what are they, pentagons, and squares and circles and the same. So even at that level, we can see that we've got 4 different taxonomic units. And in this case, it's fish. So we've got 4 different fish within that sample. At this stage, we do not know what it belongs to, but we can still use that as a measure of diversity. And particularly when we start looking at micro-organisms, we're not going to be able to necessarily identify them all. And so even just the genetic sequences can be used as a diversity metric. But ideally, what we want to do is then match those sequences to known sequences. So what's really important is having a reference database. Oops, going the wrong way. So we can then match the sequences from these blue diamonds and go, they belong to carp. And these pentagons are blackfish and galaxids and silver perch and so on and so on. And so what we get from our single sample is not just a detection of a single species, we can get an entire list of species that are present at that site. What is critical is this reference database. So reference databases are incomplete. They always will be incomplete. But what I would say is that our larger organisms, our vertebrates, are relatively comprehensive. Particularly for fish in Australia, it's pretty complete. Once we start getting into invertebrates that are a lot more diverse and a lot more cryptic, there starts becoming some fairly large holes in it. But without that reference database, we can't make that step to go what the sequence belongs to and identify it to species. Now, we're not always going to be able to identify things to species for a variety of different reasons, but that doesn't mean that data is useless. It might just mean that we can only identify it to genus level. And so we'll identify things to the lowest taxonomic level that we can. To give you an example of what that data looks like, so this was a vertebrate analysis. So we're looking at fish, frogs, mammals, birds, reptiles as well, across a range of sites in southern Victoria. This is a fairly common output that we get. So we get a whole list of species from our samples or our site. Here we've got our native species in green, our introduced species are yellow. So we can calculate a variety of metrics from that. We could look at total species richness. We could look at native species richness. We can look at the ratio of introduced to native species, which is another common one used, particularly for fish. But so we get a whole diverse range of species from that same sample. And then we can easily compare between different sites. So you can clearly see this is a lot more diverse site. So we can look at, again, those metrics. We can look at the composition of species, how that might change. And again, try to relate that to different environmental variables to understand what's driving it. Or it might just be that we want to know where those biodiversity hotspots are because they're the ones that we want to protect as the highest priority. Or conversely, we look at the ones where maybe diversity isn't as good as it should be. What sort of management actions can we employ there to try to improve that? And then we can track that over time by sampling at regular intervals to see, again, measuring how effective our management actions are. I mentioned before that we can't identify everything down to species level, particularly some of our really closely related species. They're too genetically similar to tell apart. So things like there's a couple of galaxid here that we could only identify down to genus, and even some that are at higher level that we couldn't identify. But you see the vast majority we're getting down to species level here. This is the one thing that I'm not talking about for waterways. Again, just to sort of highlight a different use. I mentioned that cats and foxes are incredible predators in our landscape. They are very good at essentially doing all the legwork for us and then leaving us some little samples. This is a project that was actually done out my way not that long ago. I don't want you to look at too much into that table. Just wanted to highlight the range of different species that we got. This was about 80 different scats I think we got from within a park area. So not only are we able to identify what the predator is, but what it's eating. And with this project, we're even able to do some genotyping of the foxes themselves. So we could identify individual foxes from the DNA on the outside of those individual scats. But just to highlight this, so from, I said, about 80 scats, we found 2 different fish, a frog, 15 birds, 13 mammals, 4 reptiles. Foxes and cats in particular are incredibly generalists. They will essentially eat anything they can get their teeth onto. They'll scavenge some animals. They'll predate on others. So you can get a really good diverse range of mostly terrestrial species from those kinds of scats. So really effective way to be able to, I guess, survey broadly within terrestrial systems, because I mentioned earlier that DNA doesn't travel like it does in aquatic systems. The other thing I want to touch on here is that I guess we're often being asked to measure biodiversity for these projects, but to date, people are very much focused on those larger organisms. Now, when we think about total biodiversity, I'm sure everyone's sort of familiar with this kind of tree of life diagram or variations of it. Most of our projects, people are still looking at fish or vertebrates. And that is a very small fraction of what is total biodiversity in an area, whether that's aquatic or terrestrial. But they're things that people are interested in. We can catch them, measure them, and ID them. And so we're used to looking at those things. Now, decades ago, we recognized the limitations of this in aquatic systems, and we started using macroinvertebrates to measure ecosystem health. And that's, again, because we could catch them, we could identify them, we can count them. And there's more of them. There's more of them than there are of our vertebrates, so it gives us a much better indication of ecosystem health, as well as the sensitivity to detect change. And we know that there are certain key species that are sensitive to disturbance and others that aren't. But that's still a fraction of what our total biodiversity is. Most of our biodiversity is actually microscopic. And the reason we haven't looked at it previously is because we haven't been able to. We now actually have the tools to be able to do that. So essentially we can use a variety of different assays and look at everything from our vertebrate level down to microscopic organisms and get a much more holistic and a much more sensitive measure of biodiversity in our waterways. And I think people are starting to understand this a bit more, that we now have these new tools and we need to look at how we can utilize them more effectively and not just try to apply new tools in the way that we've used our traditional methods. So I think we'll start to see a lot more of this starting to happen. But essentially, we're going to go from having taxonomic richness of maybe a few dozen or even 100 or so to thousands of organisms per sample. And that's going to give us much greater sensitivity to look at both spatial and temporal variations and to really understand ecosystem health at a level that we've never been able to do before. I feel like I'm missing Sorry, slides are a little bit out of whack there. Another way that we've been applying eDNA, which is probably very specific to a lot of the audience today, is something that we've called SourceDNA, where essentially we're looking at bacterial contamination and being able to quantify it, but also being able to track the source of it, both the physical source and the biological source. So what that looks like, this is an app that we've created. And so we can it's actually using a combination of our two methods, both metabarcoding and qPCR. And so we can look at bacterial loads and generate heat maps like this. They can get overlaid on a variety of different spatial layers to look at how that might correlate with your water pipes, your septic systems, to see where the origin might be. And by using sequential sampling along waterways, you can track that back to its source quite effectively. And then by looking at a range of other different species, you can then correlate that with what other species are present. So is it actually human contamination from an overflowing septic tank, or is it coming from someone's cows that are in the paddock next door? So you can use that to identify, tracking bacterial loads and seeing where those hotspots are, and how that might be diminishing over time, which is obviously pretty critical for human health and advising people using these waterways, but also understanding where that source is so you can really quickly and effectively tackle that, manage it, and consequently looking at how your bacterial loads diminish afterwards. Okay, almost at the end. My voice is starting to get a little bit croaky. So just summarizing, there's a variety of different ways that eDNA can be applied. I think every time I speak to a new group, they will pose a new question, a new way of, hey, can we use it in this way? And my jaw will just kind of drop and go, I've never thought of that before. So I guess the purpose of this is to make you guys aware of what eDNA is, what it might be able to do, so that you can then come up with these novel ways of how you might apply it. But everything from baseline data collection, and that might be as simple as, look, we need to know what species are there so that we can manage things appropriately, implement the right processes that we need to protect them or facilitate growth. Things like environmental impact assessments, so looking for threatened species, for developments and so on, and also tracking how that might be changing over time. That ongoing monitoring, I mentioned earlier, because it's a simple process, it's cheap and effective, we can do things more frequently, more often at larger scales than we have been able to do previously, which means we're getting better data and can make better decisions. One of the critical ones, and I think one of the ones that I get asked a lot about is, okay, we've implemented this management action. It's cost us $5 million. How can we quantify that it's had its desired effect? Well, because of all these reasons, we can now monitor and quantify whether it's having the desired effect. And it might take years for that to actually happen, but we're able to, again, do these longitudinal studies to understand that. Certainly a growing area that we're seeing, and we think this is going to have huge applications for, is the nature accounting process. And I'm sure we're going to hear a lot more about that in the coming years. I think those kind of ecosystem health metrics, whether that be soil or water, and starting to look at those micro-organisms as our targets to quantify and rate our ecosystem health that way. And just finally, as I mentioned, that bacterial contamination tracking. So there's a few different ways that they're being applied. You can probably think of another dozen off the top of your head. But just really wanted to highlight the diversity of how this can be applied. Getting back to, I guess, EnviroDNA and how projects might run, I guess one of the things that we really want to do is make sure that we are working really closely with the end user to get a good outcome. So, and I guess this is where my role comes in is understanding what the problems are and sort of using our eDNA knowledge matched up with your local knowledge and your species knowledge to try to come up with an effective project design. The sampling can be done by yourself or by citizen scientists or by ourselves. Those samples then come back to us for the eDNA analysis, and if required, the bioinformatics or any sort of data science that's required. And then reporting can be as simple as an output of a list of species from your site to sort of full reporting, historical records, investigations, data analysis, incorporating environmental variables, whatever that might look like. So, but I guess the remit, I guess, of EnviroDNA is to make sure that we are sort of advising and working with you through this whole process from beginning to end. So finally, for those that do want to learn a little bit more about eDNA, as I mentioned, there is a further course coming up. It's going to be run over the next 2 to 3 months. The reason it involves a couple of workshops, but what we are going to do, and unfortunately this is restricted to Australian people only because of our strict quarantine requirements, we're going to be sending eDNA kits out to everyone. So a water sampling kit for everyone that registers for this. We want you to then go out. You can choose your own sites, whether it's a wetland, a lake, a creek, whatever it might be. It might be your local waterway. It might be an area that you're working in. You can take some water samples and then send them back to us. We're going to do a vertebrate biodiversity analysis, so a fairly broad spectrum analysis of vertebrates that's in and around your waterway. But we do need to have samples back at our lab by the 5th of November. That's our cutoff date. If we don't get samples back by then, we're not going to be able to have results in time for our second workshop, which is on the 5th of December. So I do encourage those that are interested to jump on board and register for this as quickly as possible. It means we can get kits out to you and get them returned to us as quickly as we can. The first workshop is going to be in a few weeks on the 24th of October. This is going to delve a little bit more deeper into some of the technical aspects of eDNA, but also, examples of how you can design surveys to answer various questions and some of the analysis and statistics that we've done to help guide that. And then at workshop 2, at the end, we're going to be delving more into what the data itself looks like. So how it might be analyzed, how you can interpret eDNA data, because it is a little bit different. There is often grey area there that is important to understand. So we'll use some of our existing data, but importantly, we're going to go through the actual data that we're getting from your samples as well to help bring it to life to people. And I believe there is a discount code that's going to go out to all attendees today, which I think is valid for the next 2 weeks. So we want to get people to register fairly quickly for this. Okay, thank you. Thanks everyone for listening. I am getting a little bit croaky now, so I'm going to have a quick drink of water. Do reach out to me or through the water school itself. They'll put you in contact with us if you've got any ideas or questions down the track. And like I said, we're one of the things that we really enjoy doing is trying to tackle those difficult questions that you will inevitably pose to us. Right.
And we have had a few of those in the background, judging by the responses and the feedback and the interactions in the background. This is absolutely a topic that people are very involved in. And as you've heard now, this course coming up is as interactive as it gets. You get to go out into the field and we get to analyze the same data. Now, international attendees, as I understand, can still join, but you would just need to kind of look over somebody else's shoulder and you wouldn't have the kit yourself, but you will be able to see how others use it. So I would, highly encourage anybody who's interested in this science to just give it a try yourself. Don't just keep it academic. Get out in the field. And when you're looking at actual results that are that you went out and got, it's going to just bring it home a little better for you. So Emma and Halo in the background. So we'll start with Emma and then go to Halo. As you've been answering some of these questions, I guess we can't possibly get to all of them, but let's pick one or two that you think might be most relevant for the overall group, or maybe some that people had upvoted where you can tell that it's a question that a lot of people might have.
Yeah, of course. I'm going to actually throw to Halo for a couple. So there's a few technical ones that a few people asked. They were sort of repeated a few times. So one of them was following up on Peter Cook's question, which for reference, Peter Cook asked, can eDNA be used to determine how much of a particular species is present? Can you associate the type of found eDNA to the abundance of a species in the water body, or could that really just be one animal, bacteria, fungus, protist, etc.?
Well, the answer is yes. It could just be one, because our qPCR probes can be really, really sensitive. We can pick up as little as 5 copies of DNA, and there's millions of copies of DNA in one living organism. But we can also figure out relative abundances. We can never nail down exactly how many individuals using this kind of technology, but we can have relative abundances. Yes, there's heaps of this fish here, less over here kind of stuff. But yeah, it can also literally just be one that we're picking up.
And just to add to that, we've actually done that with a number of species where we've matched it up with electrofishing. So from pest species like carp to threatened species like dwarf galaxids. Your survey design does become even more critical then, because there's a lot of things that influence DNA abundance. And so you don't want to be getting false results.
Halo, anything that you had answered in the background that you'd like to highlight for the group?
I had one, I think that is good for Josh, actually, that I left open, asking, would appreciate if you can comment on sampling techniques. How many samples are required and what is the depth of sampling in water? I think that's really interesting, considering how different types of water require different types of sampling.
I would say join into our next webinar, because that will be one of the things that I'll be tackling. Look, it really depends on what you're trying to answer and what level of confidence you require. So with depth, I would say for most aquatic systems, freshwater systems, we typically just take surface water samples, within the top 10 centimeters. We are doing a little bit of work at the moment to look at benthic macroinvertebrates and whether it's more effective to get benthic samples rather than surface water. Preliminary results are saying it doesn't show much. What I would say is when you start moving into the marine space, there's not much point taking surface water samples if you're looking for benthic organisms that are 30, 40, 50 meters down. And that's when we do use things like Niskin bottles to sample at depth. So it really depends on what your question is, what your target audiences, what your target species are. And again, just designing that sampling program accordingly.
I can tell you all have a passion for this. We're about getting toward the end, so I guess what we'll do is just go around the room, and you can offer any closing remarks if you want to do that. Take that time to address any other questions that have come in. Feel free, or just anything you would want to say in preparation or to get people excited about this upcoming course. So again, back around the room here from Emma to Halo and then Josh.
Yeah, we had one question about soil sampling. So yeah, that's an area where the assays are constantly developing. There's a lot of research in that area, in that space. We do currently do soil sampling. So it's definitely something where you can do a project with us now. But yeah, there is also the benefit of this. It's constantly being researched. So you get those reference database will be added to consistently, which is nice as well.
Excellent. Halo?
I just do want to answer one more question as my closing remark. There's a lot of questions about the certainty of results, and I understand it's a new technology. People are wondering, how reliable is it? We verify all of our results using databases that use traditional methods. So usually, if we're saying this thing is here, it's because it's also been previously seen there using another method, which I think is really important to note.
Bit of a reality check. Got to have that in there. Josh, bring it home for us.
I'll probably extend on Halo's comment. I think when most people think about eDNA detections, they think about it in a very binary sense as yes or no. And in reality, it's a continuum from probably not there to almost certainly there, and everything in between. And again, these are things that we'll explore in a few weeks' time. But there are ways that you can sort of interpret your data or understand your data to give yourself more or less confidence of what those detections mean. But yeah, I think, if you've got ideas, reach out to us. We're more than happy to chat through them. And if it turns out that, hey, eDNA is not suitable, we'll also tell you that as well. So, I think as we've seen, there's a range of different ways it can be applied for a range of different organisms in a range of different ecosystems. So that makes it very useful, but also very complex to understand.
Yeah, thanks.
That's what we're here for.
Exactly. And so, do scan this barcode. If you're watching this on the computer, scan it with your phone, and you can get right to the link, or click on the link that'll come through, again, with a coupon code, discount code that'll come through to all attendees in the follow-up email. So, those who are into this field, I think a lot of times, the passion is not just for the science, but for the environment as well. And sometimes it can be disheartening to see things change. We had Bo Miles on doing a filmmaker who had highlighted some of Australia's sickest rivers, one of which they had used eDNA technology to show that it was absolutely biologically dead. We might put the links to that one there, but there are some other rivers that are recovering. And so there's good news and there's bad news out there. What we want to do is put our finger on the pulse and be able to monitor the health of this, of the river systems. And if you don't have a baseline, you're not going to be able to see how things are impacted over time. So even if nothing, even if the things that are going to happen to the river and might impact it in the future have not happened yet, get that baseline out there so that we can then see what happens, if a mining company or others come in or their development comes in, what happens to the health of that ecosystem. So do give this course a listen, and give also the there's another webinar that Helen Barclay, who's the director of the same company that we've got represented here, EnviroDNA, gave us a bit of a shorter introduction earlier. You can see that number 157. So our webinars are all numbered, so you can find them very easily. Go back to number 157. And you'll get to hear Helen's introduction into this very exciting field. So do fill out the poll results at the end. You will get this recording, and you'll get a certificate within the next couple of business days. Let us know what you want to see more of. And with that, thanks to all of our attendees, and thanks especially to each of the presenters, Josh, Emma, Halo. Thanks for being with us today. We look forward to this course with you and hope to see as many of the attendees with us in that course as possible. So with that, we'll say goodbye for now, and we'll see you next time with the Australian.